Hello and welcome back to the Deep Dive. I want to start today's session with a question, something we see in suspense movies all the time.
Oh,
You know that scene where a criminal is trying to disappear? They try to burn their fingertips with acid or maybe slice them up to wipe away their fingerprints?
right. So the police cannot identify them. It is a classic movie trope.
It is. And, in a physical sense, it might work to some extent. You can alter your appearance. You can change your face with surgery.
Yes, you can.
Exactly. But here's what really fascinated me. You can change the outside, but there is a chemical fingerprint inside every single cell of your body that you simply cannot scrub away.
That's a very powerful way to put it.
It doesn't matter what surgery you do. This internal identity is permanent.
Because we are not just talking about identification here. We are talking about the blueprint of life itself. Today, we are unlocking the secrets of nucleic acids.
This is a very crucial part of Unit 10, biomolecules.
Yeah.
If you're a Class 12 student listening to this, you know this chapter carries good weightage.
It does.
But even if you're just curious, stay with us.
Mm.
Because reading through these notes, it feels more like learning a language than just chemistry.
It really is. I often tell students that if you get lost in the formulas, you miss the beauty of it all. Nucleic acids are essentially the molecular logic of life.
The molecular logic of life, that is a big claim. Why do we give this topic so much importance?
Well, think about what any living system actually does, whether it is you, me, a tree, or even a tiny bacterium.
It grows, it survives.
It grows, it sustains itself, and most chemically impressive of all, it reproduces. It makes a copy of itself. The big question for centuries was, how? How does a parent transmit their specific characteristics, like eye color or height, to the next generation?
And that's what we call heredity.
Precisely, heredity. For a long time, that was a complete mystery, but now we know the answer lies in the nucleus of the living cell.
In the chromosomes.
Yes, the chromosomes there are made of proteins, and these very biomolecules we are discussing today, nucleic acids. They are the chemical basis that allows life to continue.
So, without this, life as we know it essentially just stops.
It absolutely stops.
Okay, let's open up this molecular hard drive then. If we zoom all the way in past the cell into the nucleus, what are we actually looking at?
So at the most fundamental level, you are looking at a chain. A very, very long chain.
And if I have to define it for an exam, the one sentence that gets me full marks.
The technical definition you need to write is that nucleic acids are long-chain polymers of nucleotides.
Polymers of nucleotides.
Correct. And because poly means many, they are also called polynucleotides.
Polynucleotides, got it. I want to visualize this. I'm picturing a long metal chain, like something on a ship's anchor.
That is a good mental image. Keep that in your mind.
Okay, but a chain is made of its links. So let us look at just one single link in that chain. You're calling that link a nucleotide.
Yes.
But this link isn't just one solid piece, is it? Chemically speaking.
No, it's not. This is where it gets specific. That single link, the nucleotide, is actually made of 3 distinct parts that are chemically joined together.
3 parts. What are they?
Part 1 is a pentose sugar. Part 2 is phosphoric acid.
Okay.
And part 3 is a nitrogen-containing heterocyclic compound, which we simply call a base.
Let me repeat that just so it's 6. A sugar, phosphoric acid, and a nitrogen base.
Correct. Those 3 come together to form one nucleotide, and then thousands and thousands of these nucleotides join up to form the nucleic acid chain.
Now, I have to pause here because I ran into a huge confusion point. I kept seeing 2 words that look almost identical. Nucleotide and nucleoside.
Ah, yes.
Are they interchangeable?
Oh, definitely not. And if you swap them in the board exam, you will lose the mark immediately. It is a very common trap.
Okay, so how do we distinguish them?
Think of it like this. A nucleoside is just the base plus the sugar. That's it. It's attached at position 1' of the sugar.
Okay, so base plus sugar is a nucleoside.
But if you then add that third component, the phosphoric acid, it becomes a nucleotide.
Is there a trick to remember that? Because side and tide sound very similar when you are under exam stress.
The trick I tell my students is in the word tide. A nucleotide has the phosphate tied onto it.
Oh, that's brilliant.
Yeah.
Nucleoside is just the side structure. Nucleotide has the phosphate tied on.
Exactly. And you must remember the position. The phosphoric acid is usually attached to the carbon at position 5' of the sugar.
Okay, so we have our links, our nucleotides. Now, how do we connect them? How do they snap together to make that long chain?
They connect through the phosphate group. So the phosphoric acid of one nucleotide links up with the sugar of the next nucleotide.
It's like they're holding hands.
Sort of, yes. The proper term is phosphodiesterlinkage, and you have to be very specific about where they connect.
Where?
It connects the 5th carbon, the 5' carbon of one sugar, to the 3rd carbon, the 3' carbon of the next sugar.
The 5' and the 3'.
Yes. That 5' to 3' connection forms the backbone of the whole structure. If you are drawing this diagram for an exam, labeling that is essential.
Got it. So we've built the chain. But we know there isn't just one type. The big ones are DNA and RNA. I feel like there's a misunderstanding here where people think, oh, it's basically the same thing, just a different name.
That is a dangerous assumption to make. Chemically, they are quite distinct. And the difference starts with that sugar we mentioned.
The S in the structure.
Right. In DNA, the sugar is sulfidity-2-deoxyribose. But in RNA, it is deodoxy.
It's all in that deoxy prefix.
It changes everything. Deoxy literally means it is missing an oxygen atom at the 2nd carbon position compared to the RNA sugar.
And why does one missing oxygen matter so much?
That small chemical difference makes DNA more stable. It's less reactive than RNA. And that is why nature chose DNA to be the long-term storage for genetic information. It is safer. So never say they are the same. DNA has deoxyribose, RNA has ribose.
DNA gets the deoxyribose. That's why it starts with D. RNA gets the ribose. That's why it starts with R. Simple enough.
Exactly.
But there's another difference in the ingredients, right? The bases.
Yes. The nitrogen bases, there are 4 options for each. In DNA, you have adenine, A, guanine G, cytosine C, and thymine T.
AGCT.
But in RNA, 3 are the same, but that 4th one changes. RNA does not have thymine. Instead, it has uracil, U.
So if I am looking at a chemical structure in a question paper and I see a U.
It is RNA instantly, no doubt.
Okay, so we know what they're made of. But let's zoom out to the big picture. When people hear DNA, they don't think of formulas. They think of that iconic shape.
The double helix.
The spiral.
Yes, the double helix. We owe that image to James Watson and Francis Crick, who won the Nobel Prize for proposing this structure. It's probably the most famous shape in all of biology.
It is. So what's the best way to understand it?
The best analogy is a twisted ladder. Imagine a flexible ladder that you have grabbed at both ends and just given a gentle twist.
Okay, a twisted ladder. Let's map our chemistry onto that. What are the rails? And what are the rungs or the steps?
This is the primary analogy you should always keep in mind. The rails, the long sides of the ladder that you would hold on to, are made of that backbone we talked about.
The sugar-phosphate chain.
Alternating units of sugar and phosphate, yes. And the rungs, the steps you would stand on, are composed of the bases. But here is the key. Each step is actually 2 bases meeting in the middle.
So one base from each side.
One base sticking out from the left rail and one from the right rail holding onto each other with hydrogen bonds.
So they are paired up?
They're paired. And they don't just pair randomly. This is the molecular logic part. They are incredibly picky.
Who pairs with him?
Adenine A always links with thymine T, and cytosine C always links with guanine G.
A with T, C with G.
Always. This is called complementary pairing, and this entire structure exists specifically to solve a problem.
What problem is that? Why go to all this trouble of pairing them up? Why not just have a single string?
It solves the problem of self-duplication. Imagine you have that ladder. If you unzip it right down the middle, separating the steps, you now have 2 separate strands.
Okay, I'm picturing the ladder splitting in half.
Right. Now, because of that specific pairing, rule A only with T, C only with G, if you have one half of the ladder, you automatically know exactly what needs to be built on the other half.
Oh, I see. If I have a strand that reads AGC, the other side must have been TCG.
Precisely. The strand itself acts as a template for its own replication. When a cell divides, it passes these identical copies to the daughter cells. That is how heredity works at a chemical level.
That is how your eyes look like your mother's.
It is all in that reliable copying mechanism.
That is fascinating. It's like a backup system built into the architecture itself.
It is the most efficient information storage system we know.
I want to take a moment here for a mental movie. Listeners, if you can, close your eyes for a second. Picture that double helix.
See the 2 strands wound around each other. Now zoom in on the rungs. See the hydrogen bonds acting like a special kind of glue.
Specific glue. A to T, C to G.
And notice that the 2 strands are not identical. They are complementary. They're opposite matches. If you have one, you have the key to build the other.
And that explains the chemical fingerprint we started the show with.
Yes. While the fingerprints on our hands are physical ridges, the fingerprint inside is the specific sequence of those bases. The order of A, G, C, and T along your DNA is unique to you.
Unless you have an identical twin.
Exactly. Unless you have an identical twin, nobody else on Earth has your sequence.
And this is what they use for DNA fingerprinting in forensics.
Exactly. It has revolutionized forensic labs. They can identify criminals from a tiny bit of biological evidence, a hair, some skin, a drop of blood.
But it's not just for crime.
Not at all. It is used for paternity tests to determine who a child's father is.
And I read in the notes it is even used to identify dead bodies after accidents.
Yes, sadly. When physical identification is impossible, comparing the DNA of the victim to their parents or children can identify them. It is also used to study the biological evolution of different racial groups.
It's amazing how much utility comes from just 4 chemicals arranged in a spiral.
It really is.
But we have talked a lot about DNA. I feel like RNA gets a bad reputation, like it's just the sidekick.
RNA is definitely the unsung hero.
Is it? Because the naive assumption is that RNA is just a backup copy of DNA. Like you keep the original in the safe and carry a photocopy around.
It is not just a copy. Think of it this way. DNA is the master architect. It sits safely in the nucleus, holding all the blueprints.
Right. The architect doesn't go to the construction site and lay the bricks.
Exactly. RNA is the workforce. RNA molecules are the ones that actually take the message from the blueprint and then go out and synthesize the proteins. They build the body.
So without RNA, the DNA is just a library that no one can read.
That's a perfect way to put it. And structurally, RNA is different too. It is usually a single strand, unlike the DNA double helix.
Not a ladder.
Not a ladder. It can fold up on itself in complex ways, but it doesn't have that long double-stranded structure.
And for the students listening, we need to know the specific types of RNA, right?
Yes. For your exam, you need to be able to list the 3 main types.
What are they?
There is messenger RNA, M-RNA, ribosomal RNA, rRNA, and transfer RNA, tRNA.
mRNA, rRNA, tRNA. Simple enough to remember.
They each perform different functions in making proteins, but for this unit, knowing the names and that they are types of RNA is the key requirement.
We have covered a huge amount of ground, from the crime scene right down to the molecule. Let's try to distill this. If a listener has to explain nucleic acids in under 60 seconds, what's the deep dive summary?
Okay, the 60-second revision.
Point 1. Core idea.
Nucleic acids are polynucleotides found in the nucleus. They are the blueprint of life responsible for heredity.
Point 2. The visual.
Think of DNA as a twisted ladder or a double helix. The rails are sugar phosphates, and the rungs are the base pairs, A with T and C with G.
Point 3. The key distinctions.
DNA uses the sugar deoxyribose and the base diamine. RNA uses the sugar ribose and the base uracil.
Point 4. The terminology trap.
Do not confuse nucleoside with nucleotide. Remember, the nucleotide has the phosphate tied on, and they link 5' to 3'.
Point 5. The application.
The sequence of bases is your unique chemical signature used in DNA fingerprinting, while RNA is the active worker that synthesizes proteins.
That is a solid wrap-up. Studying this really makes you pause. We are all walking around with this incredibly complex library inside every single one of our cells.
You really are. And I want to leave the listener with one final thought to think about.
What's that?
We talked about the sequence A, G, C, T. It is just 4 chemical bases, 4 letters in an alphabet.
Right.
But somehow the sequence of those simple 4 letters codes for every single complex protein in the human body. Your eye color, your insulin, your muscle fibers, your brain chemistry. It's all just a rearrangement of those 4 letters.
That is mind-bending. The complexity of a human being comes from a 4-letter alphabet.
It is the ultimate language.
Well, on that note, I would encourage you to open your Unit 10 textbook. Actually look at those diagrams of the double helix and practice drawing that nucleotide structure. It really helps.
Absolutely. Good luck with your studies.
Thanks for listening to The Deep Dive. We will catch you in the next episode.